So welcome everyone who are attending this webinar, and thank you for your attending as well. And also thank for those who in the future view this talk on YouTube. So today, I want to take this opportunity to discuss, I think it's a very exciting line of research that is on the crosstalk between the ubiquitin-proteasome system and autophagy or macroautophagy specifically. And this is also a field our lab had made some recent contributions to. Let me get this pointer. So let's see here. Oh. So in this talk, after a brief introduction, into the UPS, the ubiquitin-proteasome system, and autophagy. Then I will primarily discuss how does proteasome malfunction impact on macroautophagy and how does autophagic impairment affect UPS performance, and end up with some practical implications. So the UPS, I think probably everybody knows by now, this is major protein degradation pathway in the cell. Is responsible for the degradation of more than 80% of cellular proteins for two fundamental purposes. One is protein quality control. It is through targeted degradation of misfolded proteins in the cell. And the second is the so-called regulatory degradation. That is through degradation of perfectly normal but it's already done its job, no longer needed proteins. So through regulatory degradation, the UPS actually regulates virtually all cellular processes and the cellular functions. So there to study the UPS functioning or performance, a number of modified fluorescent proteins has been created that to show changes of UPS functioning in the cells or even inside the body. So these are fluorescent protein, primarily GFP, either can be carboxyl fusion with a degradation signal sequence, such as diagram cell 1, a ubiquitination signal sequence, or modified N-terminal amino acid and then fused with a modified ubiquitin. And all being used in various research. And in my lab, we primarily use this GLP-U or GLP-DGM because at a very early beginning of my career, we created adenovirus vector that can introduce GLP-U into cultured cell called myocytes. We also created a ubiquitous transgenic mouse model that overexpress GLP-DGM. And here shows that this GLP-DGM mouse responded to proteasome inhibition. Here's MG262. As expected, when you inhibit proteasome, so the GLP accumulate in the tissue. So these tools have played a great role, a very important role in showing or unveiling the UPS malfunction in the heart and other tissue and organ as well. So the other catabolic pathway is the autophagic lysosomal pathway. So for those attended the webinar yesterday, there was a very nice early career investigator, Prof XJ actually already gave the very nice introduction into autophagy. Autophagy actually consists of 3 different types: macroautophagy, as shown here, and chaperone-mediated autophagy, as well as macroautophagy. So for this talk, of course, we're going to primarily involve in macroautophagy. So I'm not going through all these detailed procedures here, but I want to just highlight that in this process in autophagy formation and the maturation process, LC3, especially the lipidated form LC3, known as LC3-2, is incorporated into the autophagic membrane at a very early stage of this formation of autophagosome, say in a figure 4, and then stay with autophagosome throughout the whole process. Those in the inner membrane of the double membranes of autophagosome actually will be degraded along with the cargo, autophagic cargoes. So the LC3-2 as well as the LC3-2 flux, you're going to hear that a lot in my later part of my talk, are commonly used to reflect respectively autophagosome abundance and autophagic flux, okay? So in terms of protein quality control, apparently the targeted degradation of misfolded protein primarily is performed by the UPS. But more recently, there are other, the chaperone autophagy, chaperone-mediated autophagy CMA also may be able to selectively degrade some of the misfolded protein if that protein with a KFERQ motifing. And of course, the aggregate proteins or protein aggregates are believed to be degraded by can be degraded by macroautophagy here. So here, as you show, these seems to be in parallel or in series, but in fact, that there are emerging evidence that the two major catabolic pathway, UPS and autophagy, do intersect or interact. That will be the topic today. And why it's important to look into that, because I'm not going to show you the data that both the impairments in UPS and autophagy occur in most, if not all, heart disease, and it contributes to the progression of a large subset heart disease to heart failure. So studying the interplay may unveil some new therapeutic targets. Meanwhile, improving proteasome functioning or coordinated enhancement of selective autophagy can improve protein quality control and protects against proteotoxicity, and is generally beneficial to the cells, enhanced proteasome or autophagy. So activating one of them may compensate for the function insufficiency for the other. Let's see if that turned out to be that way or not. So as a couple of examples to show you that UPS malfunction in vivo demonstration of UPS function insufficiency was showing illustrated these slides. This was done in desmond-related cardiomyopathy that took advantage of our GFPT gene reporter mouse. When you co-express the GFPT gene with a mutant desmin, that mutation was linked to human DRC or human desmin myopathy. You can see here there's a huge accumulation of GFPT gene indicating UPS functions impaired or inadequate in this cardiac proteinopathy mouse heart. And overexpressing wild-type desmin didn't do that. Also, in this same category, mouse model, even the same model, but the data shown here is slightly different. Is the same desmin cardiomyopathy mouse model induced by overexpression another mutant protein, misfolded protein, is arginine 120 mutated to glycine of the crystalline, the so-called R120G mouse model. And in this desmin cardiomyopathy mouse model, which we recently did a pretty comprehensive characterization in terms of autophagic flux using the LC3-2 flux, showing here that that it can show you here the early stage at 1 and 3 months. These are the time points, this DRC mouse model, the heart function still in compensated stage. And you can see the autophagic flux or LC3-2 flux are dramatically increased. But later in the decompensation stage, at 6 months, this is the time point very close to the lifespan of this transgenic mouse and R120G mice they have a a shortened lifespan. And see here the lysosomal inhibition with buflomycin A1 BFA here could not further increase LCS32 in myocardium. Unlike in control, littermate control mice still can increase a lot, but no longer increase in the transgenic R100G transgenic mouse heart. In other words, indicates that LCS32 flux is almost shut down. This, I want to point out, this is just a 1 hour inhibition. You can see in control mouse heart, there's a huge accumulation, means there's a pretty rapid flux LCS32 in normal heart up to 6 months of age, but that was undetectable in the DRC mouse heart. So at an early stage, at least, there's a coexistence of UPS malfunction or impairment and activation of autophagy. So I'm gonna try to address or discuss 2 questions in this talk. First, question 1 would be, how would cardiac proteasome malfunction impact on autophagy? So here the hypothesis is proteasome malfunction activates autophagy through upregulating P62. The preliminary support, or the reason we came up with this hypothesis was we already show you the proteasome malfunction and autophagic activation coexist in that, at least in DRC heart. So there might be, because proteasome malfunction induced autophagy. And also we saw there both at the mRNA and the protein levels in both type of both models of DRC, the P62 are significantly upregulated. So what is P62? P62, also known as sequestering 1. And here shows you a domain structure of P62. This P62 is actually uniquely positioned to mediating the removal of ubiquitinated proteins by autophagy. At least structurally, it looks like that way. Because at a C terminal, you have this ubiquitin binding domain, UBA domain, that combines to ubiquitinated proteins, especially polyubiquitinated proteins. And also in the middle here, you saw there's a LIR motif, LCS32 interacting region that can interact with LCS32. Remember, LCS32 incorporate into autophagic membrane and the feedforward at a very early stage. So that is believed to recruit this autophagic membrane onto ubiquitinated proteins. These ubiquitinated proteins are very likely aggregated because at the N terminal of P62, you have this PB1 domain. The PB1 domain, especially after phosphorylated by various kinases, that can self-dimerize or even polymerize. That property actually makes P62 help the aggregation of ubiquitinated protein is bound. So it binds to P62. Of course, there are other similar proteins in various cells as well. So empirically, I'm not going to show the data. We already reported that pharmacological proteasome inhibition, I'm going to abbreviate it as a PSMI. Pharmacological PSMI can activate proteasome, activate autophagy, in the heart, in cultured cardiomyocytes, as well as in vivo, in myocardium. And here we want, but that has never been demonstrated in any organ that with a genetic approach. So here I want to show you that with cardiomyocyte restricted a genetic inhibition, a genetic PSMI, through cardiac-specific knockout of an essential proteasome subunits, PSMC1 here. PSMC1 is a 98 subunits is required for the proteasome assembly or proteasome function. And here, as expected, when you inhibit a proteasome, there's a huge accumulation of ubiquitin conjugates in the tissue, in the cell, as shown here. So also you can say here that this is alpha myosin heavy chain driven Cray mediated condition knockouts. And you see here this significantly reduced, but not that much reduced, not completely abolished yet. It's just because this knockout is so severe, and it actually does not take, it's not until the time completely or almost completely knockout. We know that alpha myosin heavy chain Cray can mediate a pretty thorough knockout in cardiomyocytes, but in this case, actually, it's pretty. This is the area you can see here that not this. We didn't include in that image, but you can see here that there's a downregulation PMC1 and accumulation of ubiquitinated protein. But importantly, of course, there are GLPG genes can be accumulated once you introduce to those knockout heart. Very importantly, the P62 is also upregulated, increased, and LC32 is increased. The increase in autophagosome further that demonstrated by introduced the GLP LC3 reporter. You can see here in the knockout, there's a dramatic increase of GLP LC3 puncta or autophagosome are increased in this knockout heart. Meanwhile, if we double label ubiquitin conjugates with P62, you can see here the increase of ubiquitinated protein primarily in the form of aggregates and is mostly co-localized with P62 here, okay? Since that increased LC32 in the heart could be due to two potential causes. One is the increased formation of autophagosome or increased formation of LC32, or decreased removal of autophagosome. There will be two completely different status in terms of autophagic lysosomal pathway. So to differentiate that, we had to do this so-called autophagic flux assay. It's very essential to do that, to differentiate that. We turn to culture cardiomyocytes. First, start with mouse cardiomyocytes. We took advantage of the flux mice. We also the flux cardiomyocyte out of the cardiomyocyte out of the flux mice, neonatal mice, and then culture it and treat it with Cray, adenovirus-driven Cray, to knock down or knock out PSMC1, and then assess the baseline LC32 level, and also after inhibition of the lysosomal removal with bufalomycin A1, BFA. And you can see here the flux, LC32 flux is significantly increased in this PSMC1 knockout cells. But not in wild type cells. You do the same way to the wild type mouse cardiomyocytes. The flux is not a significant increase compared to control virus, beta-gal virus. We also used the neonatal rat cardiomyocytes in a slightly different way. That here we use siRNA specific knockdown PSMC1 or control siRNA against luciferase. And similarly, you can see here the knockdown increased P62, and the inhibition of lysosome further increased P62 that in the knockout cell much more dramatically than in the control cell. In other words, the P62 flux is increased in the proteasome, genetically induced proteasome inhibition, rat heart muscle cells. That point out that P62 is primarily degraded by autophagy. So P62 flux, just like LC32 flux, reflecting autophagic flux. And when we perform the immunofluorescent confocal microscopy to double label ubiquitin and LC3, you can find here that first of all, the ubiquitin conjugates is increased in the PSMC1 knockdown cells. And after lysosomal inhibition, there's a much more dramatic accumulation of this ubiquitin aggregates compared to the control cells. And these aggregates primarily co-localized with LC3, indicating that lysosomal removal of ubiquitin proteins is significantly increased by proteasome inhibition, or in proteasome inhibited cells. So we have shown the associative evidence P62 always increase, always there, associated with increased autophagy. Is P62 really required or plays an essential role in that induction of autophagy by PSMI? The answer, in vivo evidence never been reported until this one here. So we subject P62 and wild-type mouse to proteasome inhibition. In this case, we use MG262, and that also inhibit lysosome that with BFA. And again, LCS2 flux was increased by proteasome inhibition in the wild-type mice, but that's completely lost in the P62 mice. So demonstrating that P62 is required for proteasome inhibition to activate myocardial autophagy. So the next question within the question number one that we would like to address is, through what mechanism proteasome malfunction upregulates P62? Well, first of all, there are several possibilities. We thought that first, the protein stabilization, because the P62 binds to ubiquitin conjugates and form aggregates. And these aggregates, of course, no longer accessible to the proteasome. The proteasome can only degrade individual ubiquitinated protein molecules. It has to be soluble. It has to be individual. Once form aggregates, the proteasome would not be able to degrade them. That's why autophagy may have to come in to remove them. Second, because when we did an RT-PCR to look at the P62 mRNA level, actually increased by by more than 4-fold compared to control heart. That indicates the transcription or increased synthesis of P62 may also come into play. So then the next question is, what pathway mediate or what transcription factor mediate P62 increased transcription in this case? Well, our prior studies, I'm not going to show any of the data in collaboration with our Dr. Taixin Cui in University of South Carolina, that we have actually showed before the NERV2 pathway, NERV2 P62X actually will be involved in this increased P62 transcription. But more in this talk, I would want to show you that how we look into the TFEB, the transcription factor EB, which is a known master transcription regulator of lysosomogenesis and autophagy genes as well. And the list is keep going longer as well. There are other genes have been added to, but primarily TFEB is a transcription factor, can stimulate or activate the expression transcription of a network of genes The network is called a clear network, and this including all the lysosomal genesis essential genes, as well as many autophagic genes. So how cell biologists has worked out how this transcription factor is regulated, at least so far. That at the baseline, during fetid condition, we know autophagy is activated by starvation, at least. That was to help deal with energy crisis by self-digest portion of cytoplasm to provide fuel, to at least temporarily survive from energy crisis caused by starvation. And but in a fat condition or rest condition, TFEB is constitutively phosphorylated by mTOR complex 1. And the phosphorylated TFEB, it can be phosphorylated by other kinases as well, but primarily always important is mTOR1. That phosphorylated TFEB will be bound by chaperone 1433, which will sequester TFEB in the cytoplasm, prevent it from getting into the nucleus. Then, as a transcription factor in the cytoplasm, will not be able to do anything. And but under starvation or exercise, some other lysosomal stress conditions, that first of all, the mTOR probably going to in starvation, certainly mTOR will be shut down. And because of nutrient deprivation, mTOR will be suppressed. And that stops phosphorylated TFEB. Meanwhile, the calcium release from lysosomes through this nucleolipin 1 is a cation channel on the lysosomal membrane. That release calcium, and calcium bonds to activates calcineurin, the calcium carbogen calcineurin pathway. Actually, calcineurin and calcineurin serve as a phosphatase to dephosphorylate TFEB, and then TFEB, dephosphorylate TFEB now can move into or translocate into the nucleus, where bonds to the clear elements and activates the expression of a large subset of genes involved in lysosomal genesis and the autophagic and autophagy. Okay. And we have observed previously, and also report about 10 years ago, that proteasome inhibition actually can activate calcineurin NFAT pathway. So we knew calcineurin can be activated by proteasome inhibition. So that's why we thought, well, maybe calcineurin and the TFEB pathway may be involved in our here. So this is just show one of the figures here. And the NFAT nuclear translocation by proteasome inhibition is calcineurin dependent, because when we use sequestering A to inhibit calcineurin, the nuclear translocation is nearly completely blocked. So then we will just want to look into first in vivo in mouse heart, whether the proteasome inhibition, pharmacological proteasome inhibition, can activate TFEB in the heart or not. So that's easy to do. We treated mice, wild-type mice, with a proteasome inhibitor, in this case, bortezomib, BZM. This actually is the first FDA-approved proteasome inhibitor that has been used clinically to treat a couple of diseases, primarily the multiple myeloma, very effective actually. So we use that to inhibit proteasome. This is global inhibition, of course, that we can see here. The slow migrate form of TFEB-A actually reduced, and the faster migrating form increased. We know that migration actually consistent with the dephosphorylation of TFEB. And when we do nuclear fractionation, indeed, the nuclear TFEB is increased. Actually, both A and B are increased here. And also, very importantly, the representative target genes UVRAC, VPS18, MCON1, M6RP, and P62. So they're all significantly upregulated by proteasome inhibition in the heart. This is global inhibition in the mice. How about genetically only inhibit the proteasome in cardiomyocytes? This we went back to our PSMC1 knockout. As you can see here, the nuclear translocation of TFEB certainly increased because the nuclear TFEB positive nuclear cardiomyocyte a lot more in the knockout cells, knockout heart compared to control, legitimate control hearts. And again, most of the representative genes also upregulate. The TFEB target genes also upregulate. So these both pharmacological and genetic inhibition of proteasome clearly can activate TFEB in the heart. So how about in cardiomyocytes, in culture cardiomyocytes? So we turned to neonatal rat ventricular cardiomyocyte in culture. We treated with bortezomib again, and you can see how strikingly this migration difference and consistent with dephosphorylation, which is consistent with activation of TFEB. And when we do nuclear fractionation, clearly TFEB-A is dramatically increased in the nuclear fraction in cell treated by BGL. And also you can see here only that if you believe this is a dephosphorylated form, and all the nuclear in the nuclear are the dephosphorylated form. Okay, and this happened at 12 hours in the case, and we did another time for 24 hours remain the case. And the immunofluorescence staining just additional way to demonstrate the nuclear localization, and that certainly is increased. I notice my time's going very fast. I'm gonna speed out a little bit. And the targeted gene expressions increased. So that activation of TFEB by PSMI, it turned out indeed is calcineurin dependent, because if we inhibit calcineurin with pharmacological inhibitor sequestering A, that nuclear translocation and of TFEB-A is significantly reduced, and also the target gene expression is attenuated. And the bottom panel C actually shows the use of genetic means, use siRNA-mediated calcineurin A beta knockdown. They basically achieve the same result as sequestering A. This activation of TFEB by PSMI also requires mucolupin-1 or MCON1. Remember, that's the calcium or iron channel or calcium channel on the lysosomal membrane that mediate the calcium release to activate calcineurin. And this knockdown certainly blocked proteasome or attenuated proteasome inhibition, induced the nuclear translocation of TFEB-A here. Also, the target gene, representative target gene of TFEB activation by PSMI was attenuated by knocking down MCON1. So by then, so we basically deleted a pathway that MCON1, calcineurin, TFEB, P62, that this pathway participates in the mediating the activation of autophagy by proteasome malfunction. So back to the P62 knockout mouse and the wild-type mouse, we looked at preliminarily into the activation of a TFEB. And it turned out that in wild-type proteasome inhibition, with MT262 in vivo, you can see the nuclear translocalization of TFEB, and that disappeared in the knockout heart. So, and also the target gene expression by proteasome inhibition, induced by proteasome inhibition, also attenuated in P62 knockout heart. That suggests to us that P62 may play a feed-forward role in TFEB activation by proteasome inhibition, which actually first documentation, never documented before. And we don't know what exactly mechanism that, but we suspect maybe through, for example, that the mTOR suppression. And because proteasome inhibition looks like can suppress the P62, the mTOR activation, and the mTOR actually co-localize, especially during proteasome inhibition with P62 in the aggregates. So we postulated that P62 mediated protein aggregates sequester mTOR complex 1 or mTOR into that aggregate. That will sequester away mTOR from TFEB. Then not going to phosphorate TFEB, allow the TFEB dephosphorylation to go in, promoting that dephosphorylation and activation. So this is the overall model for the one leg, how proteasome malfunction to activate autophagy, proteasome inhibition, both pharmacologically and genetically, can increase calcineurin activity, that increase dephosphorylation. Meanwhile, probably through the feedback loop, that a feed-forward loop that to inhibit mTOR as well, that activate TFEB activation lead to expression lysosomal and autophagic lysosomal genes that this including mCON1, mCON1 certainly is known to feed forward here. Of course, P62 will mediating the protein aggregation and also recruits autophagy to remove ubiquitin protein aggregates. So question number 2 will be much shorter, and that will how would a cardiac autophagic impairment that could impact on UPS functioning. Here, the hypothesis is actually the hypothesis was from others, primarily originally from. I'm going to show you this piece of data here. That in 2009, Dr. David Rubinstein of the University of Cambridge published a paper in Molecular Cells shows that when you inhibit autophagy, you increase P62. Meanwhile, increase proteasomal substrates. Here showed endogenous P53 as well as this surrogate GFP. Ubiquitin, this is a G76V ubiquitin fused GFP. That's another surrogate substrate for the UPS. And they showed that proteasome activity, peptidase activity was not affected by the inhibition. In this case, for ATG knockdown, ATG7 knockdown. But if you knock out P62 or knock down P62, that accumulation of ubiquitin UPS substrates or proteasome substrates no longer occurs. So that they went on, did a lot of additional experiment and demonstrate very nicely in HeLa cells that autophagy inhibition can compromise UPS mediated degradation that in a P62 dependent manner. So what we did so far, what we've confirmed that actually happened in cardiomyocytes as well. In cultured neutral rat cardiomyocyte, we use a different way, including ATG7 knockdown. In addition to that, we also use the plasmacin L1 to inhibit lysosome, or also use siRNA to knock down RAP7, which is important for the fusion of autophagosome with lysosome. That in all cases, the P62 is increased, and very importantly, when you knock down P62, that GFPU, which is the surrogate substrate of UPS used here, their accumulation was completely abolished. So that basically confirmation. More importantly, we demonstrate for the first time that actually happened in vivo as well. So we treated our GLP-DG mice with 2 different autophagic inhibitors. One is bortezomib A1. You can show here GLP-DG, of course, will accumulate with short treatment of BGM, but a longer term treatment of BFA, and you need even more GLP-DG accumulation. That happened in the heart, also in the kidney as well, in other organs as well. I just showed 2 representative. Here, in terms of P62's role, mediating role, in vivo has not been, to my knowledge, I might have missed on the paper, but I'm not demonstrating yet. It should be done, has not been done yet. So to summarize the second one, that basically when you have, when you inhibit autophagy, or when you have an autophagic impairment, you abrogate P62, you increase P62, you at least 2 different ways. One, stable protein stabilization, because we know P62 is degraded by autophagy. Second, transcriptionally, at a transcription level, here shows that actually P62 through inhibit KEAP1, which is the ubiquitin ligase for NERV2. And NERV2, as you know, there's a master transcription factor in redox against oxidative stress. So when you during autophagy inhibition, actually P62 and the NERV2 form a feedforward loop as well. So you will increase through NERV2, the transcription will be increased. That's what it can do to the ubiquitinated protein. The P62 will hijack the ubiquitinated proteins from proteasome or on their way to the proteasome and make them to aggregates, and that will impair the degradation of ubiquitinated substrate by the proteasome. So this is the final slides, pretty much, that P62 actually is right at the center of the crossway between autophagy inhibition impact on proteasome and proteasome function insufficiency impact on selective autophagy. And on one direction, proteasome malfunction through NERV2 or accumulated ubiquitinated protein. Here, newly documented carcinogen TFEB pathway increase P62, and P62 will in turn to help promote activation of selective autophagy, presumably for removal of ubiquitinated proteins, especially in aggregate status. However, when you have an autophagy impairment, that the P62 also increased, but in that case, P62 will hijack the ubiquitinated protein substrate from the proteasome. The outcome will be similar to PFI, proteasome function insufficiency. So what? Well, apparently this, what I highlight here, provide some new magnetic insight into the crosstalk between the 2 fundamental catabolic pathways, which undoubtedly is significant in long run and at the present as well. So here I want to highlight 2 important, even practical, current, even immediately implications. One is for research. So we know some of their researchers using the changes, simultaneous increase in P62 and LC32 as the indicator of impaired autophagic flux due to impaired removal. And since, as we showed here, you can have a situation that probably going to happen pretty often in the cell is the simultaneous increase of P62 and LC32 proteins in the circumstance where the autophagy actually is activated. It's not impaired. So that caution has been taken, or actually, in my mind, it's very, very risky to use that as an indicator of impaired autophagy flux. and you have to do a flux assay. Clinically, so we know, there are probably no clinician attending this talk, but for just for the sake of information, what we found here actually helpful to help the interpretation was actually their clinic finding reports that you use rapamycin analogs instead of compared to cyclosporin as immunosuppressants in cardiac transplant and kidney transplant patients. It turned out the rapamycin is better in terms of suppress cardiac hypertrophy and improve diastolic function. And I think what we presented here or highlighted here actually offers an interpretation for that, because you use rapamycin, you will promote TFEB activation, but cyclosporin will do the opposite. And then finally, I would like to thank all the people who, over the years, a lot of collaborators, this research paper, specifically this Dr. Pan, Lee, Julie, Tian, and Wu, these are actually five first authors. And most of them actually already out of the lab. The project started 10 years ago, so that's why many of them are already out. But in close collaboration with Dr. Su Huabo, actually the project was started when Huabo was still in my lab, and now he's already tenured faculty in Augusta University Medical College of Georgia, and the people in his lab participate in this work. And this all names in blue actually are the current member of my lab. The PSMC1 flux mask was generously provided by Dr. Bedford and Dr. Meyer from University of Nottingham Medical School at the UK. And Dr. Fang plays an instrumental role in assisting us to get this P62 knockout, very crucial for this study. And a long-standing collaborator, Dr. Liu and Dr. Che already mentioned earlier, also made a significant contribution to this line of study. And currently my research is supported by this NIH grant. And last but not least, thank you all for your attention. I see whether we still have some time that I will be happy to answer if you have any questions.
Thank you, XJ, on behalf of everyone for this very interesting and exciting talk, very clean and conclusive data. Thank you. So there are a lot of questions already. So I'm going to read them out.
Sure.
So the first question from Heinrich Tegmeyer is, could you please comment on ER stress and the unfolded protein response?
Certainly, the ER stress and the UPR is probably the best studied protein quality control mechanism in the cells. But in that case, both UPS and the autophagy, especially the UPS, are essential in so-called ER-associated degradation, ERAD. And of course, initially we found UPS exclusively responsible for that, but more recently, autophagy also added to the list. In terms, if you will, the ERAD, historically ERAD just said the proteasomal UPS mediated. It's very important, and the ERAD, the UPR, and ER stress certainly is very important for heart disease as well, and some of the branches more recently has been tied more extensive study by what labs I am familiar with, like Dr. Jill Hills at UT Southwestern and Chris Glombotsky at San Diego State University. They all made a very similar contribution in that area in terms of ER stress, UPR in cardiac pathogenesis. So limited by time, I could not get into any of those.
Okay, the next question is from Mark Rennick. So you used buflomycin A1 for autophagic flux assay, but it is pretty costly. So however, chloroquine or hydroxychloroquine is not. So some papers used to prefer chloroquine. So what is your opinion on the choice of the inhibitor?
So this is from Mark. Hi, Mark. Thanks for coming to the, Mark's one of my PhD students. Thanks for the, he made a very important contribution to the lab and to the field as well. It was on the PKG regulation of proteasome. I didn't have time. It's not in this topic, so I didn't talk about it. And he's in Hopkins now. It's a faculty over there. So, this is, I'm glad you brought out this question. This is a very interesting question. The whole chloroquine, hydroxychloroquine, many people, if you didn't know before, you must know now during this pandemic age, advertised by, some of the presidents. So, indeed, there are people use that chloroquine to inhibit lysosome. It turned out actually chloroquine, as you know, now is a drug that maybe was believed to as a basic compound. It was believed to get into the lysosome and neutralize the acidic environment that will reduce, we know, the hydrolysis, all kinds of enzymes for hydrolysis of different substances, including proteins in the lysosome compartment, or require their best pH actually acidic. So, and if you raise the pH, it was supposed to inhibit the activity of the lysosome. But there's a recent paper indicated chloroquine actually, in this case, that did not appear to function that way. Actually, chloroquine primarily inhibit the fusion of autophagy and lysosome very slowly. And it differs from bufalomycin A1, of course, we knew that, but bufalomycin A1 BFA is inhibited vacuole type of proton ATPase, which is primarily on the lysosomal membrane, that you prevent the proton from pumped into the lumen of lysosome. Then you also increase the pH that will inhibit those enzymes in the lysosome. That's how it inhibits lysosome. And it's very effective. BFA is much more effectively inhibit lysosome. We have shown now we start with like 2 in vivo, 2 consecutive injection, 1 hour in between, and then at the second hour after first injection, we can see the huge accumulation in normal cell, normal heart of LC32. More recently, we optimized a little further. We found even just 1 hour, one dose. 1 hour after injection, you can see the LC32 a very discernible accumulation at baseline. So that's why we like that better. And there's a number of issues with chloroquine to be used that way, not just for COVID-19, even for the for this flux assay. I know the chloroquine is much cheaper, and bufalomycin A1 is a lot more expensive. But I would suggest, if you could, bufalomycin A1 is much better than both in vivo and in vitro. Okay, in vitro chloroquine probably is at least better than in vivo. In vivo, I heard some colleagues tell me if you want to achieve a very effective inhibition of lysosome, you have to use pretty high dose. That's already pretty toxic to the animal. And it generally, if you look at the reports already reported, the paper reported using chloroquine, they often require much longer time, say 24 hours. You inhibit lysosomal degradation of autophagy for 24 hours. Who knows what else happened there? So in this case, my philosophy would be you want to hit lysosome really hard, really quick, and for a short time, and then to see how the flux changes. So I certainly recommend the BFA. I have no commercial benefit with BFA, but not against chloroquine that way either. But and also I didn't read the paper honestly, but recently I read it from third party that said in the guideline, the Bible of autophagy, somewhere it said actually chloroquine initially may even increase autophagy flux. So that kind of makes things more complicated. So if you use our chloroquine, you saw the autophagy increase, flux increase. who knows? That's from your disease or whatever status of your experiment, or from chloroquine itself. So that for those reasons, I think the buflamycin A1 works much better. I think most people agree with me.
Yeah, actually, I agree with you. We also didn't have luck with chloroquine and went to loipeptine or buflamycin.
Thank you.
Next question is from Giulio Agnetti. So do you have a sense for what's the percentage of ATP used by the PQC system in the average adult heart? I think the total should be around 6 kilogram ATP per day. So to me, it should be a substantial amount. Perhaps increased proteostatic demand could contribute to energy starvation in the failing heart.
Questions about ATP, the requirement for UPS degradation. Certainly, this is the ATP consuming process, and in terms of ATP, there are a couple reported study. One we got involved too. That if physiological level ATP actually inhibit proteasome activity, this may be a physiological regulation, and to put proteasome activity on hold until you have stress, like early phase ischemia or other stress, you have ATP reduction, and that actually right away you can activate proteasome that way. That's just, of course, a hypothesis. But the fact is, when you have a higher proteasome, if you have a higher level, ATP show a biphasic effect on proteasome activity. Yeah, it is costly, and I had no idea how was the amount, the total in kilo to be used by the heart. But if you activate proteasome, and generally we are just says activate. You just want to recover its impaired from impairment, and that's the cost for sure.
Okay, so there are two questions from Asma Bukalfa, who thanks you for your interesting talk. So do you suggest that autophagy machinery in the heart is macroautophagy?
If I follow the question, do I suggest autophagy in the heart is macro? Is the one I'm talking about or the other one? MI?
This one.
MI or MA? Micro, MICRO or macro?
I think the question is probably whether the main mechanism of autophagy in the heart is macroautophagy.
Oh, okay.
What about other roles of autophagy, like microautophagy maybe? Yeah.
Oh, I see. Okay. So macroautophagy, the ones we commonly talk about, certainly is the best studied. It probably is the most primary ones in terms of all three types of autophagy. And CMA, chaperone-mediated autophagy, began to attract attention from the community. And it's certainly in terms of protein quality control and some even lipid metabolism that CMA may be involved as well. Micro, the direct uptake of substrate from the cytoplasm to the lysosome, that one I haven't seen much report or many reports. I mean, I don't really know. We don't really know how important that in normal or diseased hearts.
So her second question is if you observed any increase of cell volume when the proteasome is inhibited.
Yeah, we actually reported before that the proteasome inhibition for a reasonable amount of time that can induce hypertrophy, just like a tag. The one we reported, the calcineurin MFAT pathway activated by proteasome inhibition, that same paper, cardiovascular research paper in 2010. In that study, we showed proteasome inhibition can induce hypertrophy.
So the next question is from Thomas Polini-Cornelia. So he says it's a nice talk. He has three questions. So what is the effect of bortezomib on mTOR signaling in cardiomyocytes is the first one.
What the effect of proteasome inhibition on mTOR?
Yes.
Yeah, we showed a very briefly preliminary data, if you will, that data was shown in the supplement. So the protein inhibition can suppress mTOR.
And if the effect of bortezomib on lysosomal cathepsin activity, so what is the effect on cathepsin activity?
It would protein inhibition on
of BAZM.
Yeah. Okay. Lysosomal enzyme. I don't know the direct effect. If you use a specific, in our case, it's not an issue because we have both pharmacological and genetic approach employed. So genetic approach will just specifically inhibit the proteasome. In terms of indirect, certainly, we have showed in those represented genes, some of them are actually cathepsin D, cathepsin B. Those are proteases in the lysosome, and TFEB activation certainly mediate the synthesis, increase those components. It's basically all the components of a lysosome, in addition to autophagy.
So the next question is from James Jung. So have you looked at mitophagy? So he's wondering if P62 upregulation can preferentially target mitochondria for autophagy degradation.
Short answer, no, we didn't look into mitophagy. That's important. And P62 might involve in targeting defective mitochondria for the autophagy removal. And but I think there's a paper show there different cell type, different situation. This adapter protein, if you will, the P62, there's I showed you in one of the slides. There are several others. It may play a slightly different role. So at least along the same line, if not P62, would be other adapters of the same shape.
So there is an addition from Asma to her question. So she said that some papers suggest that the autophagy machinery is different from macroautophagy upon shear stress, mechanical stress, like in kidney. And she asked, what about the heart?
I don't know. Sorry. In terms of shear stress, activate different machinery. That's interesting. I'm sorry. I didn't pay attention to it.
A question from G. Lee. So thank you for your talk and really enjoyed it. Apparently, proteasome inhibition leads to autophagy activation. Your data suggests that this autophagy activation is a compensatory response to remove ubiquitinated proteins. Would you please comment on its implication in clinical situation of proteasome inhibition in related cardiac toxicity observed in cancer patients receiving bortezomib? And would enhancement of autophagy activation play a protective role in such patients?
Okay. Hi, G. G is another PhD student. So the question is a long question. Okay. The proteasome inhibitor, certainly in human in clinic, the use of first and second generation of proteasome inhibitor to treat multiple myeloma. There's a decent percentage of patients actually develop cardiac toxicity and show cardiac toxicity, even heart failure. And even though some of them, if you stop the treatment, stop the inhibitor, proteasome inhibitor, maybe some are reversible. And in terms of the activation of autophagy, I don't think it probably involved in the toxicity. If probably it may help compensate, alleviate the proteasome inhibitor side effect on other organs or non-tumor targets. But meanwhile, in terms of treating malignancy with proteasome inhibitor, the activation of autophagy certainly make it less effective. So that's certainly form a good base for co-inhibit autophagy in those oncological applications.
Okay, next question is from Francesca Sacchi. So thanks for the interesting talk. I wonder if you can comment on the role of UPS and or autophagy in sarcomere disassembly, particularly during cell cycle.
Yeah, so sarcomeric protein, there was a study show all sarcomeric protein are degraded by the proteasome. That might be overstatement, but at some portion in different state, the autophagy may be involved, especially in pathologic condition. But in normal sarcomeric, in cell cycle, in the sarcogenesis, the disassembled sarcomeric protein, when you reassemble, in the process, extra, not this stoichiometry among different sarcomeric proteins is very important. So if you some surplus of one partner protein, some of the sarcomeric proteins, say one more than the others, then it will be degraded. That is done by the UPS. Of course, a whole chaperone will involve in there as well. It's a very sophisticated process. And in terms of degradation of sarcomeric protein, people said calpain may be involved to come in to cleave those sarcomeric protein out of sarcomere first, and then UPS took over.
The next question is from Ron Vignosi. So, hi XJ, thanks for the great talk. Do you think these P62 mechanisms might feed directly into the switch from adaptive to maladaptive cardiac growth via calcineurin NFAT or mTOR, for example? So does the myocyte differentially regulate the UPS during these phases?
I'm sorry, I missed the question. Can you read it again?
Yeah, sure, no problem. So do you think these P62 mechanisms might feed directly into the switch from adaptive to maladaptive cardiac growth? For example, via calcineurin NFAT or mTOR?
That's a great question. Hi, Ron. The switch involving switch of maladaptation. I think the impairment of autophagy, yes. If you come to the point, I know in many of the disease process, such as peripheral hypertrophy, or even in our desmoid cardiomyopathy, apparently early on there's an increase of autophagy, which coincides with compensatory stage, and later the decompensation of metabolic rebounding kick in, and that often associated with autophagy impairment. Yes, in that scenario, you have autophagy impaired, the increased P62, as I showed, it will be detrimental because it can hijack the normal ubiquitin substrate from proteasome. So in that scenario, yes, I think very possible. It's interesting to design experiment to test that.
Okay, then there is a comment from Asma. So she says, I think that the stress in the heart is a shear stress via blood. That's why I asked my previous questions, and she thanks you for the nice talk.
Oh, thank you. I'm sorry I couldn't answer the question because I didn't read about it.
Well, it's
so actually, I have one question for you as well.
Okay.
Yeah, so how would you, if you look at interplayers of autophagy and the ubiquitin-proteasome system, so how would you distinguish if it's a secondary effect on autophagy or a primary effect? Do you think P62 TFEB is there like the reliable target to look at?
Okay, that's a good question. Which basic question about UPS impairment and autophagy impairment, which comes first, and because the P62 may play a different role, apparently play a different role in a different scenario. I guess first of all, you need to assess autophagic flux. It doesn't matter which one comes first. If autophagic flux is reduced, impaired, then your P62 is going to be detrimental. So, right, if it's autophagy not impaired, the P62 is probably more beneficial than detrimental. That's a simple-minded answer.
Thank you very much. Very interesting.
Thank you. You're welcome.
Thank you, XJ. I mean, I actually quite liked Asma's question about how shear stress influences autophagy. I guess if I was in the autophagy field, if it wasn't addressed already, that would be the first experiment I'd be doing. Put cells under shear stress and see if that changes anything. I suspect.
Yeah, it'd be very interesting to see how the shear stress, mechanic stress that affect. You can look at some of the already known pathways, signal transduction pathways, mTOR pathway, all kind of that related to autophagy. Kind of get some gasp, and then you form hypothesis to go ahead and test it.
Definitely.
Yeah.
Well, look, I mean, I really enjoyed the talk. Really wonderful data. I mean, you've given us a great preview because this work has just been published, from what I understand and from what I've seen.
Right, right.
And so really thank you. Great insight, and actually we get to sort of scratch beneath the surface as well, and sort of ask some really questions about the study that you've just published as well through this forum. So thank you, Sonia. Thank you for chairing. I really appreciate your help with this, and thank you for the audience that have stuck with us, for 1 hour and 20, which is great. So thank you once again, and
thank you.
Weekend.
Thank you both. It's my great pleasure, and sorry for dragging a little bit longer than I planned it.
Absolutely. You're not even close to the record. I think the record was something like 1 hour and 50 during this webinar. So you're absolutely in safe waters. Don't worry.
Okay. All right.
It was great.
Thank you.
See you everybody on Monday again.
See you.
Bye.
Take care.
Bye.